a bacterial gene regulation. Mr. Eckdahl?
Hello. Gene expression is the flow of information from DNA through RNA into proteins. It's regulated in all cells, which allows for various levels of differentiation, where muscle cells could produce a different set of proteins than, say, brain cells. The knowledge of gene expression can be applied to various fields such as health, where it could allow us to understand certain disease processes like cancer, which are caused by either a lack of proteins or an excess of certain proteins. It can also be applied to synthetic biology for the production of pharmaceuticals or biofuels. This image illustrates the start of transcription. This is a strand of DNA before the gene, which would be right over here. This section right here is the -35 region and the -10 region. It's being located by the sigma protein of the RNA polymerase before the RNA polymerase starts transcription. My research mainly deals with the -35 region because it has been shown to have a big effect on the rate of transcription. The purpose of my study was to discover the effects of mutation of the -35 region of a bacterial promoter on the regulation of gene expression. I went about discovering this by mutating the -35 region of the PTAC promoter. I replaced the standard TTG-ACA -35 region with 6 ends, which produced 4,096 random variants of the -35 region. I measured the effect that this change had on the expression of genes. I was able to conduct this research through the use of a system called pClone-Red. pClone-Red allows high school students and undergraduate students to perform promoter research without the need of expensive machinery or extensive knowledge. It functions through the use of BSA-1 sites on a left-facing promoter, which cut the left-facing promoter out, and it can then be replaced with a right-facing promoter, which in this case is my mutated promoter with the 6 ends in the place of the -35 region. It's glued into place through the use of a ligase buffer, so it can then produce red fluorescence protein. My overall method was to design and order the mutant promoter DNA, then clone the DNA into pClone-Red so it would be inserted into E. coli. I then plated the E. coli onto plates of LB plus AMP, and then I picked the clones from the plates so I could isolate individual -35 sequences for analyzation. I measured the fluorescence of those clones through the use of fluorimetry. I sequenced the DNA of the clones so I could link fluorescence to individual sequences. I analyzed the data. This is an example of one of the plates that I would have used to pick colonies from. You can see the green is caused with Golden Gate assembly, which is the process of replacing the left-facing promoter with the right-facing promoter, fails. You can see these 3 different shades of red are caused by different efficiencies of the transcription process, which in this case is caused by different sequences in the -35 region. This is 2 more examples of the plates I used to pick clones from. I wound up picking 81 clones for RFB measurement and DNA sequencing. This is a list of all the clones that I picked. You can see the clone number, the sequence of the clone, and the specific -35 sequence, as well as the red fluorescence of the clone. The red fluorescence is expressed as a ratio of the clone's red fluorescence to PTAC's, the original PTAC promoter's red fluorescence. You can see that a couple of them were actually stronger than the original PTAC, which I found surprising. Here I've highlighted a couple high efficiency promoters, medium efficiency promoters, and low efficiency promoters. And the mean RFB expression ratio of my clones was 19.7, and the median was 7.3. Most consensus sequences are developed by analyzing just the top 5 to 20% of promoters. But I wanted to analyze all of the clones that I had gathered and all of the sequences I had gathered. So I developed a novel weighted consensus method. The way it works, each For each position, each base in that position is assigned a score based on the RFP of the clone. So for example, this clone 65 here, the G in the first position of the -35 region will have a score of 128, and the T in the second position would also have a score of 128. But down here at clone 75, the A in the first position would get a score of just 1.5, and the A in the second position would get a score of 1.5 also. I summed the scores for each base in each position, and then divided by the sum for each base by the sum of all bases for that position. And that created this image, which illustrates the p-clone -35 consensus that I developed. I decided to make 4 variants: G, A, T or C, A, T or A. Here is my p-clone -35 consensus compared to the published -35 consensus. You can see that they are pretty different. They are very similar. A couple of reasons for these differences. My sample size was only 81 promoters, whereas the sample size used to create the published consensus was many more, almost every promoter in the E. coli genome. The published consensus was developed by just comparing the frequency of the bases in that position, whereas I used my novel weighted consensus method. The sequences used to develop the published consensus were all natural and found in E. coli, so they shared ancestry, and so they weren't purely random, whereas mine were all synthetic and entirely random. My sequences were all surrounded by the same PTAC promoter context, whereas each of the different sequences in E. coli would be surrounded by entirely unique promoter contexts. From here, I was interested in this just shows my 4 variants of my consensus sequence as it compares to the published consensus. You can see that 2 of them actually function better than the original consensus, but 2 of them function worse. And you can see, or less efficiently, I should say, you can see the mean of my variance was 99.0. From here, I was interested in discovering what effect the sequence context of the -35 region would have on the efficiency of transcription. And so I used this sequence, which was published by DeMay in 2007. And I wanted to create 2 promoters which were as different from the original and from each other as possible. And so this W, it could stand for either an A or a T. And in pSym1, I decided to put an A there, but in pSym2, I decided to put a T there. And then I inserted my 4 variants into both pSym1 and pSym2, as well as the published consensus, and I created this table out of that data. You can see that in pSym1, my 4 variants function similarly to what they did in PTAC, but in pSym2, there was a decrease in efficiency of my variants. The mean of my variants in PTAC, again, was 99, but the mean in pSym1 was 85.8, and the mean in pSym2 was only 20. This image illustrates the sigma factor of the RNA polymerase interacting with a major groove of the -35 element. This inspired me to create models of both one of my 4 variants of promoters and one of my 4 variants in my consensus sequence and one of the least efficient promoters out of my 81 that I had gathered. Here in the variant, you can see this positive lysine interacting with the negative phosphate group, and the negative glutamic acid interacting with the amino group of that base. And you can see the neutral valine interacting with the methyl group of that base. Over here, you can see that the positive lysine still interacts with the negative phosphate. However, the negative glutamic acid has nothing to interact to because of the different sequence, and the neutral valine also has nothing to interact to. I should point out that model is really simplified, and not all the amino acids are there, and they aren't in the right places necessarily. A couple conclusions I drew. Sequences that are different from the published TTG ACA still function and function similarly. Some sequences actually function better than the original TTG ACA. 81 different promoters yielded the new p-clone -35 consensus. The 4 variants, all 4 variants of my p-clone -35 consensus function at some level. The sequence context of the -35 region matters more than I initially thought it would. My set of 81 promoters is valuable to the synthetic biology community for applications ranging from medical to energy to environmental. My 81 promoters and the 4 variants in my consensus sequence were submitted to the Registry of Standard Biological Parts, which was established at MIT in 2003 and is the largest and most widely used collection of parts. It has over 7,000 parts, and it has both physical clones and sequence information. This is a screenshot from my page on the registry. Here's some selected references, and I'd like to acknowledge Dr. Jay Myers, my high school teacher, and Dr. Todd Eckdahl, and the undergraduate research students in the Missouri Western Synthetic Biology Lab. Thank you.
Thank you.
We have 5 minutes of questions and answers, please. You stumped everybody, Mr. Eckdahl. Any questions for Mr. Eckdahl? Thank you, good night.